Okay, hi everyone. My name is Chen Xing, and I am a technical support at Norwegian Europe, and I will be the host of this webinar. I'm so delighted to see so many of you were able to join us today. Today, my colleagues, product marketing manager Sony and the product manager Lin Qing will go through uncovering cellular complexity with spatial and single-cell transcriptomics. But before that, let me briefly introduce our company. So Norwegian was founded in 2011 and has been growing rapidly since then. We are continuously developing our portfolio of NGS and the third generation sequencing. And today we are present with services in various fields of science, from research, oncology to food and water testing. Norwegian is a global company with local contact persons and sequencing facilities in Europe, Asia, and the US. We have over 2,600 employees and more than 5,600 customers. Our first European sequencing center is located in UK in the Cambridge Science Park. The UK lab is equipped with Illumina technology and our patented intelligent sequencing platform called Falcon 3. The automated workflow of Falcon enables us to decrease turnaround time, minimize the factor of human error, thereby yielding consistently high-quality outcome, leading us to process large amounts of data. Additionally, we have launched another NGS lab in Munich, Germany. The DE lab is becoming more operational and with both NovaSeq Plus and the BioReview sequencing facilities. We provide a full range of next generation sequencing services. For example, like human, plant, animal DNA sequencing, various types of RNA sequencing, metagenomic services, and epigenomic services. There are many reasons why you should choose Norwegian as your sequencing partner. Providing consistently high-quality services is our priority. To achieve this, we perform multiple quality control. There are sample QC, library QC, and data QC. We have a large capacity, which we are increasing all the time. Recent example of this is opening our German lab facility and using our patented Falcon 3 technology, which has not only a positive impact on quality and the capacity, but also on turnaround time, since Falcon can run 24 hours for a full day within a whole week. We also offer customizable end-to-end solutions. This means we can process your project from extraction to analyze data, but you can also send us extracted DNA, RNA, or pre-made libraries and choose to only receive raw data. Our product manager team and the labs are continuously optimizing protocols and services to ensure we have a sequencing solution that fits your needs. Lastly, our multiple sequencing facility equipped with Illumina, PacBio, and the Oxford Nanopore technology give us a largest sequencing capacity in the world. We are very proud to support researchers worldwide in their work, and today have a clear record in the scientific community. We have been cited in the over 16,000 research papers with a total impact factor of more than 4,090. We are also driving the NGS industry forward with more than 60 NGS-related patents. Your contact persons are present in almost every European country. Get in touch with your local representative today to discuss NGS solutions for your projects. You can contact them directly or reaching out via our website. We also regularly host seminars in universities and various institutes across Europe. So if you would like us to organize a seminar near to you, please do not hesitate to get in touch with us. And I would like to briefly introduce our talented European sales team. They are based in different regions across Europe, like England and Ireland, Germany, Southern Europe, Nordics, Eastern Europe, and Benelux. Our European sales team functions as your first person of contact technology. So please do not hesitate to contact us with any next generation sequencing related requests. So thank you for your listening. That's all from my side for now. Now I will hand over today's topic to our product marketing manager, Sonny. So Sonny, it's your turn.
Thank you, Chengsun. Give me a minute to set everything up. I hope everyone can see my screen. Good morning or good afternoon, everyone, depending on which part of the world you are joining us today from. Without further ado, let me get right into the topic for today's seminar, which is uncovering cellular heterogeneity using spatial and single-cell transcriptomics. Here is a quick overview of what we will talk about in today's seminar. I'll start with an introduction to transcriptomics and the different transcriptomic solutions we offer at Novogene. I will then move on to spatial transcriptomics, which is one of the focus points of today's seminar, under which we will talk about the different kinds of spatial technologies available to the research community, as well as the applications of spatial transcriptomics and what kind of research questions can benefit from making use of this modern transcriptomic technology. We will then talk in details about 10x Genomics, Visium technology that we employ at Novogene as part of our spatial transcriptomic service. I'll then guide you step by step through our service workflow, starting from sample requirement, going all the way till the accompanying bioinformatics analysis. And then I'll hand over to Ling Chen, our product manager from the headquarters, who's looking after the single-cell transcriptomic services. He will walk you through the background of single-cell transcriptomics, the evolution of the single-cell transcriptomics technologies over the years, the application of this modern-day transcriptomics method. He will then talk about the steps of the workflow, starting from sample all the way up to generating the data. He will also touch upon our bioinformatics analysis support that we have available for you to easily obtain publication-ready figures from these large data sets. And then he will also share with you case studies, which are research projects that use Novogene-powered single-cell transcriptomic solution to obtain high-quality data. This will give you an idea about our experience with running such projects globally, as well as provide you with an insight into what kind of research applications can benefit from this service. With this, I will move on to the introduction for the talk today. I think all of you will agree when I say that biology is a fascinating field. And as you can see here, it's built on different levels of complexity, starting from the smallest unit, molecules, which form macromolecules such as DNA, and different macromolecules come together to make up organelles, such as mitochondria, which exist within cells. And then cells come together to form tissues, which then many tissues come together to build organs. Organs together make up an entire organism, and finally, multiple organisms form a population. And understanding how these different units interact with each other is fundamental to understanding the molecular mechanisms behind different complex biological processes. Many of the modern day transcriptomics technologies, such as single cell transcriptomics, that's one of our topics for today, allow us to zoom into individual cells, helping us to identify cell-cell variability, rare cell types, and how different cells respond to stimuli. Even more exciting is spatial transcriptomics, which is the other topic for today, which not only reveals the gene expression at a single cell level, but also retains the spatial context, showing us where the different cells are located within a tissue or an organ and study their corresponding gene expression profiles. Studying biology and biological processes while zooming into at the different levels that I talked about in the last slide can be complicated, and this is where omics technologies can be helpful. So omics refers to the study of totality of something. In the context of biology, omics technologies are employed for probing and analyzing large amount of data representing the structure and function of a class of biological molecules of a given biological system at a particular level. And this has substantially revolutionized our methods that we use for interrogating biological systems. So the output of omics technologies, these are named based on the particular biomolecule level, followed by the suffix omics. So for example, transcriptomics, this is the study of the set of transcripts, and this is at the center of today's seminar here. Many of these omics technologies are such as genomics, transcriptomics, or epigenomics. These are primarily next generation sequencing based these days. Some of the other technologies, such as proteomics technologies, the assays from Olink, for example, these also rely on next generation sequencing technologies. And simultaneous study of each of these omics can provide us a more accurate, holistic, and representative understanding of the complex molecular mechanisms that underpin biology. So transcriptome, which is a complete set of transcripts in a cell or a group of cells under a particular condition. For example, these could be looking at different time points during a drug treatment or different physiological conditions, or maybe a specific developmental stage. And transcriptomics, this is the study of the transcriptome and is most commonly employed to profile gene expression, which is the mRNA content of the cell. One of the contemporary methods of studying transcriptomics is using RNA sequencing. This is a sequencing technology that uses next generation sequencing to provide us information about the presence and quantity of RNA in a biological sample at a given moment and analyze the continuously changing cellular transcriptome. The advantage of this technique compared to traditional methods such as Northern blot or microarrays is that it's not limited to pre-designed probes. It offers high sensitivity, has a high dynamic range, and it also helps us in detecting low abundance transcripts, isoforms, and subtle changes in gene expression. The applications of RNA sequencing can be divided into two broad categories. One is the characterization of the transcripts to figure out what they are. The other application of RNA sequencing is to be able to look at gene expression in a quantitative manner. And here I have listed some of the research areas and applications that can benefit from transcriptomics. However, this is in no way an exhaustive list. With the dropping prices of both short and long read sequencing, RNA sequencing is being employed across diverse fields of science. And keeping in mind these diverse applications of RNA sequencing, we offer a very extensive range of services for different RNA species, such as mRNA-seq for mRNA, circular RNA-seq for circular RNA, and so on. We also offer what we call as total RNA-seq that can provide you with a comprehensive picture of the interaction network between different RNA species. Our full-length RNA sequencing services on the PacBio or Oxford Nanopore Technologies platform can be useful for researchers interested in alternative splicing or discovering new isoform. For microbial researchers or researchers working on infectious diseases, these services could be of potential interest. For today's talk, we will focus on these two services here, the single-cell transcriptomics that helps us look at gene expression profile at a single-cell level. At Novogene, we are excited to now start to offer end-to-end single-cell transcriptomics service powered by 10x Genomics, starting from sample preparation, single-cell library preparation, sequencing, followed by data analysis. We also support pre-made library sequencing for all the different leading single-cell analysis kits on the market, such as those from Parse Biosciences, Scale Biosciences, and so on. We also support single-cell assays such as single-cell ATAC or single-cell CRISPR screening for pre-made libraries. And the next service is spatial transcriptomics, which is particularly useful in getting a tissue organization level gene expression profile. Now let's get right into the details about this service. I'll get started with the background. So I started out my talk by saying biology is complex. Just as no man is an island, a cell's behavior and function are not isolated, but rather influenced by neighboring cells organized in a three-dimensional space to form a tissue or organ. So for example, cancer growth and progression, these are spatial processes that involve the breakdown of normal tissue organization, invasion, and metastasis. When it comes to neuroscience, spatial information is important for questions related to disease progression or of different neurodegenerative diseases or why certain neurodegenerative diseases affect one part of the brain but not the other. Similarly, during embryo development, spatial organization is crucial for organogenesis during embryo development. And this is exactly why spatial transcriptomics finds its place in many of these research areas where learning about gene expression profile in the context of spatial organization of cell and tissue architecture is important to learning about the biological processes. So studying tumors in a spatial context can help advance our understanding of how complex crosstalk between cancer and surrounding non-cancer cells evolve. Similarly, it has been a helpful tool for obtaining insights into progression of different neurodegenerative diseases, discovering biomarkers, and developing personalized immune therapies. Another research area that has benefited and continues to benefit from spatial transcriptomics is developmental biology and helps in elucidating gene expression profiles during organogenesis or embryogenesis. Very broadly speaking, there are three ways to profile transcriptomes while preserving spatial information. The first one is sequencing based methods, the second one is probe based methods, and the third one is imaging based methods. The classification is made based on what the final readout of targets is and how spatial contents are obtained. So sequencing based methods, these rely on whole transcriptome sequencing based on spatially barcoded DNA. Probe-based methods require counting of barcoded probes of known targets, whereas imaging-based methods depend on repeated imaging cycles to obtain the final readout of known targets. At Novogene, our spatial transcriptomics service is powered by 10x Genomics Visium technology, which is a sequencing-based spatial transcriptomics technology. Another example of an imaging-based spatial transcriptomics technology is 10x Genomics Xenium. And if you have projects that require Xenium solutions, please contact us so that we can discuss what solutions we can offer there. So today, for the spatial transcriptomics part of this talk, we will exclusively focus on 10x Genomics Visium technology, and we will delve into more details of this technology in the next couple of slides. So 10x Genomics Visium platform provides tissue morphology information via staining and imaging. While FFPE is a common tissue preservation method by researchers, it damages the RNA, which complicates transcriptome level investigations. And 10x Visium spatial gene expression overcomes these challenges and is compatible with FFPE samples. And these could be FFPE blocks or FFPE tissue sections. This is a probe-based method that targets the whole transcriptome in mice and humans. It enables the discovery of gene expression profiles across the tissue sections without the need to select regions of interest. Let's look at what do we need to be able to perform spatial transcriptomics using Visium from 10x Genomics. So the 10x Genomics Visium can profile whole transcriptome for any organism that uses polyadenylated mRNA, but this is only true for fresh frozen samples. On the other hand, tissues that are fixed with formalin and embedded in paraffin wax, that is FFPE tissue blocks, these often have degraded mRNA that lack a poly A tail. And this is where Visium employs gene-specific probe sets, and currently the probe sets are available for humans and mice, making the technology compatible with FFPE samples from mice and humans. And we at Novogene are currently offering end-to-end workflow for spatial transcriptomics of FFPE samples from mice and humans. The next thing we need to be able to study spatial transcriptomics using 10x Visium are Visium site-assist spatial gene expression slides. So the Visium cytosis slide shown here consists of two active capture areas, each of which are 6.5 millimeter by 6.5 millimeter in dimension. There is a second option of 11 millimeter times 11 millimeter for larger tissue sections or larger blocks. The two capture areas, they are identical to each other, and each capture area contains about 5,000 individually spatially barcoded spots. Each of these spots is 55 micron in diameter, and the space between the spots is 100 micron, which is the center to center distance. The 55 micron spot size is what gives Visium its resolution. And depending on the tissue type, about 1 to 10 spots on average will sit on top of each of these spots that are separately spatially barcoded. Now, each of these spots is printed with millions of oligos that serve as the mechanism for capturing and barcoding RNA molecules through a workflow we will discuss in details very soon. So here you can see a cartoon of one of these oligos. Let's look at each of these individual pieces that make up the oligo and the role these pieces play in the spatial transcriptomics technology. So from right to left, we have the poly-DT sequence here that is necessary for capturing the poly-A-tailed mRNA, and we will discuss how the poly-A-tailed mRNA is synthesized for these FFPE samples. Then we have the UMI or unique molecular identifier. So UMIs, these differ between every oligo to uniquely label each original transcript. They are also used to collapse PCR duplicates. Then we have spatial barcode, which is fundamental to the spatial transcriptomics technology. Every spot on the capture area has a unique spatial barcode. So sequences with the same spatial barcode can be grouped together as coming from the same spot. And at the end, we have the Illumina sequencing primer binding region that will be used for generating the sequencing library, which we will see soon. The next thing, we need to be able to spatially resolve mRNA across a whole mouse or human FFPE tissue section is a transcriptome probe kit. So the FFPE transcriptome probes are designed to against the target conserved regions of the protein coding transcriptome of mouse and human. So let's look at the RNA capture principle that is employed here to overcome the issue of degraded RNA in FFP samples that are typically without a poly-A tail. So the Visium cytosis slides, they utilize a new and specialized chemistry, which they refer to as the RNA templated ligation approach, that allows for sensitive and specific RNA detection in FFPE samples. The human and mouse transcriptome probes are split into right-hand side and the left-hand side that ligate upon hybridization to the correct target. So using this split two probe chemistry reduces non-specific signals over a single probe approach. And this introduced synthetic poly-A tail allows for the capture via the poly-DT sequence that's on the oligos. So for humans, 18,000 genes are targeted, and for mouse, more than 20,000 genes are being targeted, which allows for studying the whole transcriptome in these two species. The next important part of the spatial transcriptomics technology is a site assist device. This facilitates the transfer of the ligated transcriptome probes from the glass slide onto the capture area of the Visium cytosis slide. So we are talking about spatial transcriptomics here. So of course, having correct spatial information is one of the highest priority, and cytosis minimizes mislocalization and thus maintains the spatiality. So we just looked at the different components of running a spatial transcriptomics experiment with 10x Genomics Visium technology. Now let's look at the step-by-step workflow leading from sample all the way to data analysis. And here I'm showing you a schematic of the 10x Genomics workflow starting from sample preparation, imaging, to data processing and visualization. The workflow starts with imaging the tissues for cell morphology visualization, and this step involves preparing glass slides with tissue sections, which is followed by deparaffinization, staining, and imaging. The probe panels consisting of the split probe pairs are added to the decross-linked tissues, and together the probe pairs are complementary to the target RNA, where they will then hybridize. The hybridized probe pairs are then ligated together via a ligase to form the full-length ligation product. The tissue section on the glass slide is then transferred to the capture area using the Visium cytosist, where we take a bright field image of the tissue section. The ligated product with the poly-A tail will then be released from the target RNA using RNase digestion, and following the digestion, there is tissue permeabilization, which will allow the full-length ligation product to be captured in these capture areas via the synthetic poly-A tail that was introduced. So once this has occurred, we are then ready to generate the sequencing library, which is then sequenced on the Illumina sequencing platform, generating sequencing data, which can then be processed for downstream analysis and visualization. I know that this whole process can appear quite complicated. However, do not worry. Our end-to-end workflow will help you at all the steps, starting from sample preparation all the way till data analysis. So what do you need to do in order to opt for our spatial transcriptomics service? So as I said, you can send us the sample, which can either be FFPE blocks or FFPE tissue section. As part of our quality control steps, we assess the sample, and we will get into the details of it later. If the sample passes our QC standards, we will do the library preparation for you. We perform our sequencing on the latest NovaSeq X Plus and then proceed to data analysis if this is what you want us to do for you. In terms of what we need from you, the service is currently available for human or mouse samples, and this is because the transcriptome probe panels are designed against the genome of human or mouse, and this is what is currently available from 10x Genomics. So one sample type that we can accept from you are the FFPE blocks, and this is a preferred option, and we recommend that these blocks are no less than 3 years old. The other option is that you send us FFPE tissue section. Here we recommend a thickness of 5 micron, but thicknesses of 3 to 10 micron are also acceptable. We recommend that you prepare these tissue sections on glass slides freshly before shipping. So being able to receive good quality and quantity of data output is dependent on the sample quality. And this is why at Novogene, we have quality control steps in place before we proceed to preparing the sequencing library. If you send us FFPE blocks, we will prepare the tissue section and then stain with the HND stain and then image to determine the tissue morphology and tissue integrity. If you send us tissue sections, we then request you to send us a couple of extra tissue sections on which we can run these staining and imaging quality control steps. So RNA quality in FFPE samples plays a crucial role in the data quality, and this is why assessing our RNA integrity and quality is important. We measure DV200 to assess the RNA quality and integrity. So DV200, this is a parameter that measures what percent of the fragments are greater than 200 nucleotide. For our spatial transcriptomics service, we recommend a DV200 of at least 30% to be able to proceed to library preparation, which means 30% of the RNA fragments are greater than 200 nucleotide. And low DV200 can significantly impact library quality. And this is when we do not recommend you to proceed to library preparation or proceed with the risk. If you send us FFPE blocks for sample QC, a small number of paraffin sections will be collected for RNA extraction, and then DV200 is assessed using fragment analyzer. If you are sending us tissue sections, we also request you to send us FFPE scrolls alongside so that we can perform RNA extraction from the scrolls and then assess the DV200. If the samples pass the QC, we proceed to library preparation and end up with a sequencing library. This is the final library structure that you see here. It begins with P5 and ends with P7, which are adapters necessary for binding the DNA to the flow cell. The I5 and the I7, these are sample indexes which are introduced during the sample indexing step, and these help us to keep track of sequencing reads coming from the same sample for downstream analysis. The 10x spatial barcode, this gives us information about the spatiality, and allows us to map all reads back to a specific location in the tissue. The UMIs, they differ between every oligo to uniquely label each original transcript and will be used to count ligation events. And then we have poly-DT sequence that was used to capture a ligated probe insert. So the read one is used for sequencing the spatial barcode and UMI, which are together 28 bases. The read 2S is used to sequence the ligated probe insert, which is 50 base pair. We sequence the library on NovaSeq X Plus with a pair N150 sequencing strategy, which means that we are sequencing the library from either direction and we read 150 bases. This covers the sequence of our interest, and for each capture area, we recommend a data output of 50 gigabases. Now we'll move on to how we analyze the sequencing data that we obtained from the spatial transcriptomics experiment. So what does the Visium data look like? First of all, we have the bright field image that gives us information about the morphology of the tissue at very high resolution. The other part of the data is the gene expression matrix, and these dots here represent where we have captured RNA. Each spot represents an average expression of a mixture of cells, and together put these two data make what we know as the Visium data. So here is our standard bioinformatics analysis workflow. Our standard pipeline includes both the space ranger analysis as well as the SOIRAT analysis, which is one of the standard analysis pipeline for spatial transcriptomics. We offer analysis of differentially expressed genes, and once the list of differentially expressed genes is available, we also assign biological functions to these differentially expressed genes using some standard databases such as gene ontology or KEGG or Reactome database. That brings me to the very end of this spatial transcriptomics part of today's talk. I focused on the workflow of 10x Visium technology. Now 10x has a newest technology called the Visium HD, which is a high resolution technology. And here you can see comparison between images from Visium and Visium HD. You see that Visium HD offers subcellular resolution, and this is thanks to the design of the capture area. Unlike the Visium, where there are 5,000 spots, and each of these spots is coated with oligos, on the Visium HD slide capture area, we have continuous lawn of oligos, which is responsible for this high resolution. With this, I will hand it over to Lingchen, who is our product manager from the headquarters, who will give you an introduction to single cell transcriptomics, which is our newly launched service that we are offering in the German lab. Over to you, Lingchen.
Thank you so much, Sonny. Can everyone hear me and see my screen?
I don't see your screen yet.
Do you see it now?
No, not yet. I see that you're sharing, but I don't see it yet. Could you?
Can you see it now?
Yeah, but not in the
Yes.
presentation mode yet.
Okay. Can
Yeah,
you see it
now
now?
I can see it.
Okay,
Yeah, now
great.
it's all good. Okay.
Sorry, everybody. So, hello, everybody. I am Ling Qing. I'm from the headquarters of Novogene, and I'm a product manager in charge of single-cell RNA sequencing products. So today I will introduce the single-cell RNA sequencing service we provide recently in Europe, in Munich. Today I will talk about this part in the next 4 parts. So the first part, I will show you the background of this technology and convince you why we should use single-cell RNA sequencing on some of the researches. The second part, I will show you what we do from taking your sample and to all the way to generating the data. And the third part, I will show you our powerful data analysis ability. And the last part, I will show you some of the project experience we have for Novogene Global. Now, without further ado, let's get into the main part. So why should we use single-cell RNA sequencing? You must all be very familiar with the earlier version of RNA sequencing, which is the bulk RNA sequencing. The bulk RNA sequencing only measures the average gene expression over a population of heterogeneous cells, which means if you send us a sample, a tissue, you can only get the overall amount of RNAs of different types within that tissue. However, using the single-cell RNA sequencing, you can distinguish each cell. You can analyze the tissue in a single-cell level, which will bring you more information, and you can study the gene expression profiles of individual cells derived from homogeneous or heterogeneous populations. Recent years, there have been so many major applications and so many good papers published on the top magazines. One of them is studying the cell atlas. As you can see, from 2021 to 2022, there have been about 5 top papers published on top magazines, and most of them are studying cell atlases. Some of them study the human cell atlases, some of them focus on human immune system, and some of them study the mouse and pig and rice root. If you don't want to look at the whole picture and you want to specifically look at some specific disease or cancer, you can also use single-cell RNA sequencing. For example, if you have a tumor sample, you can run the single-cell RNA sequencing service, and we using 10x Genomics machine and generating single-cell RNA level data, and you can analyze the different expression genes among different cells. And by combining with canonical markers, you can have all the informations you want, and maybe finally decipher the resistance mechanisms and design therapy. Apart from studying cancer, you can also use this technique to study diseases, such as blood disease. You can study onset and treatment mechanisms of blood-related diseases. Also, you can study virus infection diseases and study the damage caused by pathogens to the body and immune mechanisms of immune cells within the body. If you don't want to study blood disease or a viral infection, and you want to study a healthy organism, you can also use this technique and localize the lesion size and explore the underlying mechanisms. So there are so many options for you to discover. And finally, you can also use this technique to study developmental biology. There are 2 examples. One of them are using this technology to study multi-endodermal organ atlases, and organoid models were used to map human development. The other one has unraveling the different cellular states in the mouse brain during development. Now, cell has been one of the smallest unit within human body, and our human beings have been interested in this little units for such a long time. Starting from 1665, Leeuwenhoek has invented microscope, and we for the first time see the cell from our eyes, and we're convinced that the cell does exist. And in 1965, actually, a cell sorter was invented, and it has again revolutionized the field. People has finally realized there are so many different cells and have a little touch about the heterogeneity of different cells. And from 2009, there has been a lab level invention of the first high throughput single cell RNA sequencing. And from that time on, there have been so many different technologies trying to make this result more stable, more consistent, and more cheaper. And in 2016, 10x Genomics have come to the world, and suddenly revolutionized the field again. People using this technology to study all kinds of different major applications, as I said before. And from that time on, there have been so many different technologies surrounding the 10x genomics. For example, we can use the single-cell RNA-seq to do high-throughput CRISPR screening. For example, the spatial transcriptomics is kind of based on the single-cell principle. And there are so many different techniques following this single-cell RNA-seq. Now, what we can do in Novogene, and if you want to use these powerful techniques, what you need to do and what we can bring to you, I will talk about this part. So the overall workflow of single-cell RNA sequencing from a frozen tissue using the 10x genomics is below. So you only need to send us the sample, and we input the sample using the 10x genomes machine to gene generation and run a library construction procedure. Then we use the Illumina sequencers to run through the library sequencing, and then finally have the data. If you want, we can also analyze the data for you, and there are so many options for you to choose. So I have talked about single-cell RNA sequencing as one technology, but actually this technology can apply to two major different sample types. One is, of course, the single cell, and the other is the so-called single nuclei. So why do we need single nuclei? First of all, there are frozen tissues. For your convenience, sometimes we froze the tissues and store them for a while. So this type of tissues is more easily to acquire. And the other reason for using single nuclei is the following three. If you require a completeness of cell atlas, for example, for tissues such as heart, brain, kidney, liver, and fat, if you use single cell RNA sequencing, you may lose some of the cell types. The other reason is some of the cells have large size and diameters, and they will actually stuck in the 10x machine. So in that case, we also need to use single cell nuclei RNA sequencing. And for the last part, some cells are very sensitive. For example, neuron-involved sample. And if we run single cell RNA sequencing, it may have stress response and duress impact on the sample. So that's why we recommend you using the single nuclei. The overall single nuclei procedure is like I showed here above. Basically, we took the frozen tissue and tissue dissociation and clean up the debris and run through several centrifugation steps. And finally, we have the single nuclei resuspension. And I have talked so many times about 10x technique. So why do we use 10x? So 10x is basically one of the most trending technique in the market now. It has the most reproducible results, the best results, and automated workflow and scalability. In Novogene, we use the Chromium iX platform, and let me introduce you this powerful technology. So basically, 10x have basically we put an aliquot of gel beads and an aliquot of your sample, the single nuclei suspension, and put them in the mechanism in the machine, and using by running into this lane, they will form a GEM, gel emulsion. And in every single gel emulsion, we have one cell and one bead, 1-to-1 ratio. That's the ideal circumstances. Of course, some of them have only the beads, and probably some of them have only the cell. But those data, we can remove them from the bioinformatics. Now, when they form this 1-to-1 ratio GEMs, in each single gel bead, there are millions of different small sequence binding on the surface of the bead. And in each sequence, there are TrueSeq read 1, 10x barcode, UMI, and a poly-DT. So these three parts are the most important part, and I like to explain to you. So the 10x barcode, in every single gel bead, all the 10x barcode are the same. And since there are 1-to-1 ratio to the cell, each cell are marked with one 10x barcode, only one. So that's how we distinguish every cell in the end in the NGS data. And the UMI here, UMI when in every gel bead, in every single primer binding to the surface of the gel bead, they have totally different UMI, so that we can use this UMI to quantify the transcripts. And for the poly-DT, we use that to capture the poly-A in the mRNA tail. So that's the overall procedure and mechanism of this powerful technique. And here is an example of what happens when you run through this 10x genomics capture procedure. You will have a cDNA purification, and we will run through a QC, and you can see the mRNAs are distributed like this. That's the length of the mRNA. So overall, our Novogene service workflow, let me conclude that for you. So you only need to give us your frozen sample, and we will perform the nuclei isolation from frozen tissues. Then we will run through a sample QC. We will feedback the cell quantity in nuclear suspension, the cell viability in nuclear suspension, and the nuclear morphology by examining through the microscope. And then we will perform the single cell library preparation step, which contains gene generation and barcoding, which isolating each single cell, and then post-gene RT cleanup, and the cDNA amplification and library construction. After that, we will have the library QC, and in that procedure, we will examine the small fragments, adapter rates, peak position, peak shape, to ensure that this sample can outcome good NGS data. And then we will use NovaSeq X Plus for the sequencing, and we run through P150 strategy. The recommended sample, the data quantity is 100 to 150 GB per sample. Then we will have a data QC and show you the NGS data quality, and single cell data quality. Then we will have a lot of different bioinformatics analysis waiting for you. Now let's move on to the bioinformatics part. For this part, if you want Novogene to provide you the bioinformatic analysis, we have two options. The one is cell ranger only analysis, which we will generate from the raw data and runs us a few quality control for your data. For example, read alignment, expression quantification, and quality control metrics, basically using the software single cell ranger. And if you want to dig more to your data, we can also provide you standard analysis service. So basically, after the cell ranger only analysis, we will keep going and identify the highly variable genes, cell subpopulation identification, and marker gene detection, and some functional analysis. Now, I'd like to go through our report so that you can see what kind of result you will get from Novogene service. So basically, at the first, we will show you the NGS results, quality demonstration. This is, I think, no difference than other NGS service. And then we will show you some unique things about single cell results. For example, how many cells are estimated in your sample, and the median genes are how many genes are estimated per cells, so that you can have a rough idea how many data you can use from this sample and how good are the data. And later, this part is for the cell ranger only part, this is where we end. We'll only show you the estimated number of cells and median genes. And if you want to dig further, Novogene have our own workflow, and we will run through another data quality optimization, which will filter out these 5 key factors. First one, we will filter out the cells with less than 200 genes. The second one, we will filter out genes with non-zero counts in at most 3 cells. The third one, we will filter out more than 8,000 feature count potential multiplets, which you can also see in this picture. Some of the cells are actually doublets, which means there are 2 cells and 1 bead in every in that GEM. And fourth one, we will filter out the proportion of feature count attributable to mitochondrial genes. The last one will filter out the feature count attributable to hemoglobin. And after that, we have a certain workflow to identify the highly variable genes using this formula. And after identification of the highly variable genes, we have basically all the informations to run the cell subpopulation identification, which is also within the standard analysis. So finally, you will have this most important picture, and we will run dimensionality reduction and visualization of all the cell. You can have, for example, 16 different clusters, and each one clusters represent a different cell subpopulation. For example, maybe this is T cell and this is dendritic cells. And after that, we will continue to dig in by using these 16 different clusters and by identifying those highly variable genes within those clusters. Here are some nice pictures that we can present in the results. And after that, we can go even further and using geo enrichment analysis, KEGG enrichment analysis, and reactome enrichment analysis, which are the 3 most popular enrichment analysis in the market, to study further about the function of those genes. And finally, we can also present functional annotation of transcription factor and even study the protein-protein interaction network analysis. Now, if you want more, we also have a lot of customized analysis, but you probably need to ask about what exactly you want. And please feel free to talk to our FAS. These are the list of customized analysis we can provide. So I have talked a lot about what we do, and actually, as you may probably understand, that we only started the single-cell RNA sequencing service in Europe recently. Actually, we have started this service a long time before. In China, we started this service in 2017, and so far we are actually the best in China, and we have performed more than 40,000 cases of project experiences, that including various tissue types of human, mouse, normal, and disease models, husbandry mammals, poultry, aquatic animals, plants, reptiles, amphibians. In total, we have studied more than 100 species and thousands of tissue types, and we have contributed to more than 350 publications for single-cell RNA sequencing service. Here, I'd like to show you one example of the most recent published paper by using our single nuclei RNA sequencing. So the customer sent us the frozen mouse liver and tissue tumor samples, and we performed all the procedure for him. And recently, the paper was on the preprint version. So audience, so the customer was in US, and yes, we also have this service all around the world in US and in Singapore. And I think that's all I'd like to show you about our single cell RNA sequencing service. Now, I'd like to make a summary about today's webinar. So we talked about these two products, spatial transcriptomics and single cell RNA sequencing service. For the two different products, we can accept certain sample types, and I think you can go through this page and find the information you want. If you have any more questions, please feel free to ask us now. Now we are taking the Q&A session.
Okay, thank you, Lin Qing. Thank you, Sonny. It's a very nice presentation. So let's start with the first question. The first question is, how do spatial transcriptomics and single-cell sequencing complement each other in uncovering cellular heterogeneity, and in which scenarios would one be more advantageous than the another?
Okay, I can try to answer, and maybe Lingchin, if you have something to add, feel free. I think that's a really good question, and it's becoming increasingly common to use these two technologies in complement to each other for different research questions. And as we said during the talk, the single cell transcriptomics technologies is particularly useful for discovering rare cell types or new cell types, identifying different marker genes or using marker genes to identify different cell types. Whereas spatial transcriptomics, it can give us an idea about how or where in the tissue these cells are located. So it is common to, once somebody has discovered newer cell types, to figure out where these interesting or novel cell types are located in a tissue. And this gives us biological relevance about why these certain cell types are located in certain tissues area, and also understand the developmental mechanism or maybe drug discovery or drug mechanism to know why certain cell types are targeted by certain drugs and only in certain areas. What is the tumor microenvironment, for example, that where certain cells are expressed but not others. So yeah, there are actually quite a few articles in the literature that use these methods in complement to each other. And I think the second part of the question was about when is one more advantageous than the other. I think for this, it really comes down to what is the question that the researchers are trying to answer. And if they are looking to understand the tissue organization or how the cells are organized with respect to each other, this question can only be answered with spatial transcriptomics. Whereas if they want to know only about newer cell types or cell-cell communication, this is where single cell sequencing is sufficient. Yeah, maybe you have something to add on this, Linchen.
I think you pretty much covered everything. I'd like to add one point. Based on my experience in China, most of our customers usually test their sample with single cell RNA sequencing first, and then go further into the spatial transcriptomics, so that maybe they have a basic knowledge of what their samples are, what kind of cell types are inside, and how many of them are in each different cell types. And then they will go further into spatial transcriptomics to dig into a deeper question. Yeah, that's all. Thank you.
Okay. Okay, thank you guys for your detailed reply. So the second question is Vision HD technology from 10x also supported?
Yes, we are offering Visium HD as well. But once again, this is only for mouse and human FFPE samples. And as I said earlier, and that's because we are using the probe-based technology from 10x, and these probes are designed against the human and mouse genome. So yeah, we are supporting Visium HD projects, and we have experience with a lot of different kind of tissue sections for mouse and human samples. So please get in touch with us if your projects require Visium HD.
Okay, so the next question is if 10x Genomics Visium Spatial Transcriptomics can be applied to FFPE tissue microarrays.
That's a good question. Just for the audience, so tissue microarrays, these are paraffin blocks that contain cylindrical cores that are extracted from different tissues. And as I said, the Visium or Visium HD, both of the capture arrays, the capture areas, these are 6.5 millimeter times 6.5 millimeter or 11 times 11 millimeter. And for many different kind of tissue sections, these capture areas can be large. So oftentimes, many researchers are interested in combining multiple different tissue sections on one capture area. And this is where tissue microarrays can be particularly advantageous, because by making one section from a TMA block, one can look at many different regions of interest in a single capture area. And this is a cost-effective way. So yeah, in general, these are difficult samples to work with because there are many regions of interest within the same block. We at Novogene, we have experience on working with TMA blocks. So generally for a 6.5 millimeter times 6.5 millimeter, we would recommend like a 2x2 TMA array. If you have a larger array like 3x3 or 5x5, this is where we would ask you to go for the larger capture area of 11 times 11 millimeter. So yeah, if you have projects with TMAs, do get in touch with us. We have experience with processing such samples.
Okay, thank you, Sonny. So let's go to the last question. The audience want to know how many cells in one sample can be sequenced, or how large of the frozen tissue can be used in one sequencing?
I think I can take this question. So normally we aim to capture about 10,000 cells for your sample. So that basically, if we want to make sure that we have 10,000 cells in the end, probably you will need 50,000 to 100,000 cells at the first stage, and the sample requirements for different tissue types are quite different. Maybe you can specify your tissue types, and we can talk about that later. And there is one other thing I need to mention is because the 10x this technique is not as stable as you think. If we aim to capture 10,000 cells, in the end, it will have about 50% 50% off target. So probably the final result will be around 5,000 to 15,000. So yes, we cannot guarantee the exact number, but we can provide you with a range of cells you will get in the end.
Okay. Okay, thank you. Thank you, Sonny and Lingqing. I think today our Q&A session was ended. Thank you all of you for your time.